Hello, everyone. Thank you for joining us for this next presentation in our preclinical track. In this session, James Clary will talk about the translational modeling of relapsing mouse models to inform the regimen selection in tuberculosis. Before he gets started, a couple of housekeeping remarks. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for the follow-up. You may ask questions via the Q&A tab on your dashboard at any time during the presentation. If you need any technical assistance, please use the chat tab to place your questions and comments. Before we get started, we do have a quick question, a poll question. What percentage of time do you use modeling and simulation to design or analyze preclinical studies? And we'll give you 25 seconds to respond to this poll question. See the answers coming in. So we'll wait another 5 seconds to give you all a chance to respond. And let's see. Okay, well, we have a pretty new audience. About 62% of the responses were 0 to 25%. Nearly 40% were 25 to 50%. So hopefully Jim will have a chance to convince you to start utilize modeling and simulation more. Go ahead.
All right, well, thank you, Vier. I'm glad that I'm able to talk to you all today about this topic, particularly given the poll responses. I'll be talking about this translational modeling of the relapsing mouse models in tuberculosis, particularly when it comes to regimen selection. So just to give a bit of background, tuberculosis really continues to remain a global health challenge. Last year, in 2019 alone, we saw an estimated 10 million cases. And if you read the global tuberculosis report from last year, that number is really only had expected to go up. Because despite emerging treatments and these increased investments in research and new regimens and new drugs, we're seeing that we're still not quite reaching target milestones, particularly if we look down at these two plots down here. We can actually see this is the 2020 mark, and we've actually 2019 and to 2020, we've actually increased in the number of TB deaths and mortality rate. And part of that potentially is partly due to COVID-19 setting us back even further from our goals. We see that there was a decrease in the amount of cases reported, which leads to less treatment. And so ultimately, it increases these values away from our milestones. We have a partner organization. I'm sure a lot of you have heard of the Bill and Melinda Gates Foundation, and they're a major supporter of efforts to improve the treatment and outcomes for patients with TB. And Simulations Plus is partnered with them specifically with the goal to help develop more effective drug regimens for tuberculosis with their pan-TB working group. Which we help to support quite often, and a lot of that support comes through these relapsing mouse model studies. So what defines like the relapsing mouse model? So we have mice that we give some infection to and allow them to grow tuberculosis, and then we have a few different treatment durations where we then allow recovery, and then we sacrifice to assess bacterial burden. And from there, we can determine, maybe if the slide goes, yep, we can determine the proportion of mice that relapsed at that particular time point for different regimens. And then using simple statistical calculations such as a chi-square test or Fisher's exact test, we can compare across those regimens at those time points. So we also see that these are highly influential in our regimen prioritization for further studies, and often they will inform our regimen selection for the clinical evaluation. So, these are kind of cornerstone studies that we see in tuberculosis. So, um, what we were kind of interested in, and what my partners at Simulations Plus we've kind of asked is, does this answer some of these questions that we need to be asking? And the answer is really no. When we look at just statistical comparisons at 1 month from one regimen to another, it doesn't really tell us how long we need to treat with the regimen to achieve a high probability of a cure, say like a 95% cure rate, or also, answer the question of how long does it take to get to X% relapse versus for a novel regimen versus an established regimen like HRZE. And so really we got to start thinking about rather than proportions, probabilities. And so to that extent, we've developed a model that kind of culminates quite a few of these RMM studies, and then uh, estimates intercept and slope values that allow us to calculate these key metrics of like time to 50%, time to 10%, time to 5%. Um, Recently published in AAC. And it's been really valuable as we think of how we inform RMM study design. So, as part of a TB related collaboration with Bill and Melinda Gates, a large series of RMM studies had been proposed by a partner organization. And that's kind of the case study we'll look at today of using modeling and simulation to help determine the effectiveness of a trial design. And, these are quite significant investments, the number of mice, the number of time needed to conduct these trials. And so we wanted to use our model that we developed to look at the proposed design for a variety of different regimens, both historical and hypothetical, to determine key metrics. And then also, if that design wasn't really sufficient, maybe recommend alternative designs that maximize the informativeness and decrease the overall number of mice utilized. Um, and so, when we think about like how can we approach that, uh, simulation estimation becomes a very efficient way and a very a good way to evaluate different designs over large numbers of trials without having to then perform those trials. So we took different regimens, both real and hypothetical. We looked at covariate values, which I'll show our model in a bit uh, to mimic real world targets for the study. We looked at the variety of sampling time points, both used historically in RMM studies, as well as what was proposed by the partner organization, as well as just looking at, how many animals we used over the entire study. And then we also took our model and we were able to randomly sample from our variance covariance matrix for the intercept and slope values to create new studies that we could evaluate for the different given designs. Um, and so this is our statistical model. It's a mixed effects logistics regression model. And we have study level covariates for on intercept and slope. One thing to note is this model in particular is based on our publication, but there some updates have been included as well. This model we continue to improve and update, and as we get new data in the model, we just add data to the model and continue to update these parameters. And one of the things I wanted to note here particularly is some of the data we now had 7 out of 7 dosing data, where predominantly the dosing data had been 5 out of 7 days. Um, and so now we have this covariate that we can also assess 7 out of 7 dosing, which was day dosing, which was important for the partner organization for their RMM analysis. So we simulated out 1,000 studies, drawing from our variance covariance matrix, and then assigning individual mice relapse by drawing from a random binomial distribution. And then we would then pool each simulated study with the historical data, and just like we would a new study, and re-estimate the model for the historical controls, and then as well as the new regimen that were being proposed. Um, so again, like I said, we were now looking at 7 out of 7 uh, day dosing. And we had one single BPAL control arm, which was what was intended to be used by the partner organization. And then we also created 5 different hypothetical regimens that we uh, spread out between, a very effective to less effective regimen than BPAL. And some of them we tried to, particularly looking at like BPAL and Regimen 2, we tried to put pretty close together as well to determine the design's ability to differentiate between different regimens' effectiveness. Um, and so these were our input um, values for the simulation. Slopes can be utilized to calculate these T50, T10, T5 values. We are also working on an IMAX type like model that allows us to directly estimate the T50 value and for these different regimens as well. So for the different design attributes, we evaluated a number of time points, as well as the number of mice per time point, as well as total mice for the study. And we fixed covariate effects to 4.5 CFU per mil per the inoculum, and the baseline bacterial burden to 7.5 CFU per mil. And this was according to the real world targets for our study. And so then our simulations were done in 2 stages. First, to look at the study design proposed by the partner, and evaluate that design. And then we then looked at different additional designs to determine if we could better utilize the mice, as well as fit within some logistical considerations that the partner had. So when we first looked at the design by the partner, we also wanted to look at the typical RMM study design that had been used historically, as well as a design proposed uh, by a paper from Murick et al., and some modifications of that design. And then finally, we also had a design where we had a large number of mice, which was our high-end benchmark design, and that was kind of our best case scenario, but also feasibly really impossible when you think about that we would have to have 1,500 mice to run that study. And so we looked at particularly the partner's design, which was 3 different time points with 50 mice per time point at the 1.5, and 2 months, which utilized 45 mice. So I think not surprising to anyone, when we look at the high benchmark design, we see that the values line up very well for all the different regimens. We have very narrow confidence intervals across the board, and this was what was the best that could be achieved with 5 to 10 times more mice than the next most regimen and increased time points. But again, this isn't really feasible in the real world. Um, but it gives us something to compare than our proposed design by our partner organization. Um, and we see there that the design performs reasonably well for regimens that are see most of their effect less than 2 months, which makes sense given that we have 3 time points with the latest time point being 2 months. But the confidence interval around those is quite large, and particularly for regimens 1 and 2, we don't get a very good estimation. Of note, BPAL is well estimated, and that's due to kind of anchoring that into the historical data that we have available as well. So we do see that the control arm is well estimated, even though it's very similar to regimen 2 in most aspects. And then when we look at what has been used historically, the typical RMM design, as well as the Erasmus design and its kind of modifications that we've made, uh, a few things we took away from it is that we really see that more frequent sampling results in the similar performance of a typical RMM design, but we can utilize less mice. 50% less mice in the Erasmus design with 10 time points compared to the typical RMM design with 4 time points. So we saw that, we see slight improvements with modification of number as well as consideration of spacing of the time points. And that was something, that we took and kind of ran with when we were looking at some alternate designs as well. And I think this is, kind of the takeaway message from that first part of the analysis is when we want to go to rank ordering of the results, the different regimens, particularly based on the T5 value, we see that the proposed, these are heat maps where um, we have the simulated rank order on the left and then the true rank order on the bottom. And for our original proposed design, nothing is really above 60%. The highest we get to is about 55% of the regimens are ranked correctly. Where we see in our high end benchmark, we do see some switching of BPAL and regimen 2, which are quite close together. And they have a range of the T5 of about plus minus 2 weeks. So we still see there is some swapping there. But most of the time, about 97% of the time, it orders it correctly. But then we do see for the typical RMM design and the Erasmus design and modifications, there are some improvements that we're making, particularly in design 3 and design 4, even over the typical RMM and the Erasmus design, where we see about 54% are ordered correctly, and a higher percentage of regimens 4 and 3 are also ordered correctly as well. So that made us kind of think, how could we then improve this design? So we looked back at particularly regimens 3 and 4 that worked really well. Um, and we said, what if we concentrate and add a few more mice per time point for these particular time points of interest, particularly, um, you know, either 1 to 4 months or half a month out to 4.5 months. So we increased 1 time point for each of the designs. So starting with design 7, we just increased those number of time points. So we increase the amount of mice per regimen by 5 as we go down the design, adding time points on the end to see if that increase the value of the information that we were able to get from those designs. And what we saw is, yes, it did. Particularly design 8 is what I want to draw some attention to. So here we see that for just based alone on T5, we see that it's able to assign the correct order more than 60% of the time. And that's an improvement even over the design 7, as we just add one more time point, and much improved over the original design. Um, Another takeaway is when we compare design 9 and design 10, you know, there is some minor improvements, but overall, we kind of reach the maximum efficiency with that design 8. As a good trade-off between the amount of mice that we're using, the amount of time points that we're utilizing. Even when we just add one or two more time points, we're not gaining a lot more information. And you know, because these are simulation estimation exercises, we also know the true value, and so we can calculate, what was the bias that we saw in all of these different simulations that we ran. And we saw that, design 7 through design 10 performed quite well over the proposed design, particularly when we look at 8 through 10. They're all within a 2-week interval from the true value. And we did see there was kind of a negative tendency that we get some underestimation by about 1 week of the true T5 value. But that was something that was consistent across all designs except for the high-end benchmark. So what that kind of told us is that the only way we would get around really this minimal bias is just by kind of adding a lot more mice than what we would really need to get fairly accurate estimates of these T10 and T5 values for comparison. Um, so ultimately, we proposed design 8 as it kind of gave us that best trade-off between number of mice and number of time points. And we saw that it performed better than the proposed design, but also performed better than the typical RMM design, as well as the Erasmus and design number 7. So again, that kind of gets us to that suggestion of a benefit of increase by that one more time point to allow for some better estimation. But then we appear to diminish our gains as we go from 7 time points upwards of 8 to 9 time points. And so, when we look at the simulated values from the recommended design, what we see is we were able to capture all the different regimens quite well, compared to the proposed design, which wasn't able to really capture those without significant effects prior to 2 months. And that's really, again, due to that information increase in the number of time points. Even with regimen 5, you can see we're only at the 1 month, but we're still able to capture that quite well. There's a bit of bias, but overall it's quite well estimated. And so, you know, the conclusion is really that application of these model-informed drug development tools and techniques were really critical to assessing a key non-clinical study that was going to be utilized and allowed us to make recommendations to meaningfully improve the design. And overall, we were able to decrease the number of animals utilized in this study by over 20% versus the originally proposed design, um, promoting our better animal stewardship in our R&D efforts. And as we continue to refine the model and evaluate, we can continue to increase the information we get from these designs. So I know we're running out of time, so I just want to make a brief acknowledgement to my coworkers, Al and Jessica, and all the work they put into the model and simulations, and then Dave and Deborah at the Gates Foundation, as well as the labs where we received all our information and the Critical Path Institute as well. So, yep.
Thank you, James, for the very informative session and presentation. I think we do have a couple of minutes for at least one question. How does the inclusion of a control regimen assist in the evaluation of the design performance?
Yeah, that's a great question. So, when we look at the controls arms and we have these different designs, when we include the historical data, we can see overall, no matter what design we utilize, we see that we're able to estimate those control arms, and that gives us an anchor for those study level random effects. And that's really important for the model to be able to um, use that anchor to see, what the eta for an intercept or slope for a particular study might be. What we see when we don't include those historical data is even with quite a few uh, points, we actually don't see that information being utilized as well because you don't have any real value of how far from the true model estimated value you actually are, because you only have one study, so you lose a lot of that information. So it kind of centers our true estimated value. I can't hear anything.
Sorry, I was double muted. Yeah, we don't really have time for more questions, but all I was saying was thank you all for joining us for this session. The webinar has been recorded for playback, and it will be available on our website and the YouTube channel. This concludes the session. Please head over to the lobby for the next presentation. And looking forward to seeing you in the next set of talks.
Thank you all.